All right, we're going to continue our exploration of Chapter 6 here, looking at cells. So here we have Concept 6.2. Eukaryotic cells have internal membranes that compartmentalize their functions. So one thing to consider here is there are different levels of complexity to cells. One of the first levels comes into this idea of internal membranes, right? So prokaryotic cells do not have internal membranes, but eukaryotic cells do. So we should probably stop and take a look at the differences between prokaryotic and eukaryotic cells. Now, um, there are two entire domains of life, bacteria and archaea, which have an incredible amount of diversity and only consist of prokaryotic cells. Everything that we think about as in terms of animal and plant life falls into the eukaryotic category. So there's actually a lot of creatures out there that kind of fall below the ability for us to see, but are responsible for most of the work in the world. All right, so basic features of a cell. We have some type of plasma membrane that is separating the inside from the outside. We also have this semi-fluid substance called a cytosol. Okay, it is very jelly-like with all of the stuff dissolved in it, and it is loaded with proteins. We also have chromosomes. This is the DNA that carries the genes. And then we have ribosomes floating around that make the proteins from the genes. So looking at the more simple end, looking at prokaryotic cells, something to consider is number one, it has no nucleus. Right? So here, all of the genetic material is just out in the open in the cytoplasm. Now, something to consider is this doesn't mean that there is no organization within a bacteria or a prokaryote. There actually is quite a bit, and the backbone of that organization is actually largely the chromosome itself and where the genes are located. So if you look at the internal structure, what you'll first notice is most of the DNA is coordinated to particular regions within the cell. This is something that we call a nucleoid, but unlike our cells, it is not wrapped in a membrane. You'll also notice that prokaryotic cells do not have any internal membrane organelles, so everything is happening within here. And the cytoplasm itself is only surrounded by a single layer of plasma membrane. Now, these cells can be highly organized and pseudo-compartmentalized based upon how proteins fit together, where the genes are for those particular proteins. You can see particular regions of cells that carry out particular jobs. So I don't want you to walk away thinking that these cells are not organized. They just aren't organized with membranes. So eukaryotic cells have a few differences, right? So number one, the DNA is in its own separate membrane-bound compartment, and we call that the nucleus, right? So prokaryotic cells don't have the nucleus. All their DNA is in a region called the nucleoid. Eukaryotic cells do have these membrane-bound organelles, which breaks up various functions into certain regions of the cell that are completely separated, right? Prokaryotic cells do not have that, but it still has organization. It's just not within a membrane. There's cytoplasm in between these different plasma membranes and the nucleus. Um, and in uh, prokaryotic cells, the only membrane present is the one that wraps around the outside. And interestingly, sometimes there can be multiple wrapped around the outside. Now, one of the key things here is eukaryotic cells are generally much, much, much larger than prokaryotic cells. All right, one of the key features is the idea of this plasma membrane. Going back to chapter 5, remember that we have these phospholipids. So it has two hydrophobic tails and a hydrophilic head. These can line up tail to tail to form this lipid bilayer. And this is what separates us from the outside world, right? So here we have the outside of the cell, inside of the cell. Keep in mind that this is just a layer of fat, just like that little oil and water container. If you shake it up, it pretty much just falls apart. So there's something else holding this membrane in place, and that's typically proteins. Now, you might start to think about, well, single cell organisms do quite well. Why don't we just become one bigger cell as we get more complex? The issue comes down to surface area. So here I have some cubes depicted. Here is a cube of 1 by 1 by 1, whatever unit we want that to be, it doesn't really matter. There are 6 surfaces, right? So each surface is height times width. So each surface has a value of 1 surface area. The internal volume is height times width times length. So there is 1 unit of volume. So the surface to volume ratio is 6. Well, let's see what happens as we make this bigger. Let's make this 5 times bigger, right? So here, the surface area is going to be 5 times 5, or 25, times 6, because we have 6 surfaces. That comes out to 150. If you look at the internal volume, so 5 times 5 times 5, that comes out to 125. So surface area divided by the total volume comes down to 1.2, right? This is actually the reason why we break ourselves up into many, many, many little cells. Because essentially here, the surface area is too small to allow all of the nutrients to get in from the outside to nourish something of this volume. So a much better way is to break it up into a bunch of 1 cubic cells and build the larger cube out of smaller cubes. Here, if you look at the size of, or the surface area of all these individual cubes, it's 750. The total internal volume is still 125, but we can maintain that high surface to volume ratio, which allows each one of these cells to have enough surface area to absorb all of the nutrients that they need. So the metabolic rate, or the metabolic requirements of a cell kind of sets the upper limit of size. The surface area to volume ratio is critical because if it gets too small, then a cell can't get all of the nutrients it needs to live, right? So as a cell increases its size, it proportionally goes down with surface area. So the way to solve this problem is to make something bigger from a bunch of smaller things. All right, let's take kind of a brief overview of what a eukaryotic cell looks like. Remember, one of the key features is that we have internal membranes that organize different regions of the cell into various organelles. These organelles have particular functions. Now, when we start looking at plants and animal cells, most of The internal features are the same. There are some things that are specific, and I'll try and point those out. So here's an overview of an animal cell. Number one, we have the chromatin material, that is the DNA and proteins that wrap it up. And so all of the genetic material is here. It is surrounded by a double layer of membrane. That is something that we call the nuclear envelope. And together, the chromatin and the nucleus wrapped up, um, or the, the DNA, the chromatin, and the nuclear envelope together gives us something that we call the nucleus. There are other membrane structures extending out from this nuclear membrane, and you can see here they are actually part of one system. This is called the endoplasmic reticulum. It comes in two varieties, the smooth ER and the rough ER. And this produces a large network of internal folding membranes that we can utilize to help package and sort proteins. Now, we have a further layer called the Golgi apparatus. This is physically separated in space from the endoplasmic reticulum. This is where we can essentially take proteins and shunt them to do even more complex work with them. And this is actually a series of flat pancakes that are separate units. So if we need to shunt proteins into a different location to carry out particular biochemical reactions, we can do that by passing through the Golgi apparatus. Here we have the mitochondria. The mitochondria is a double layer membrane structure, and it is where its job is essentially to produce ATP from various fuels, and it generates that ATP, and then we store that ATP to use later. We also have things such as flagella that might give us movement, or we might have various things called microvilli, which increase surface area to help absorb. We have things called lysosomes, which are kind of the stomachs of cells. We can keep all of our harsh proteins that can break down various matters in here. That way we can protect the inside of the cell. We have various things called the peroxisomes, which carry out particular oxidative reactions. We'll talk about what that is much later on. And then within the cell, we have a whole range of different proteins that form various filaments, and this produces a cytoskeleton. Remember, this plasma membrane, which wraps around the outside, is essentially a layer of fat. And that fat, if we were to shake it up, would just fall apart without some type of structure underneath. So here's kind of an overview of an animal cell. Let's take a look